Today, I'm going to talk about my research, which focuses on an important and interesting topic. In this presentation, I'll explain the main idea, why it matters, and the key points we should understand. I hope you find it informative and engaging. Imagine a child's brain cancer cell fighting to survive chemotherapy. Today, we are discussing medulloblastoma, an aggressive pediatric brain cancer. While chemotherapy is a standard treatment, it works by increasing reactive oxygen species (ROS) to kill cancer cells. However, this high oxidative stress can also damage healthy cells, leading to severe side effects. We believe that adding thymosin beta-4 to chemotherapy can increase cancer cell death through mechanisms beyond just oxidative stress. Our objectives were first, to examine how TB4 influences AAT signaling, which is a survival pathway, to measure changes in ROS during treatment, to assess the link between oxidative stress and cell survival. In our study, we used DAOY cells and D458 cell lines. We used four main things in our experiment. DMSO, this is our control. It helps us compare treated cells versus untreated cells. Cisplatin and vincristine, these are the two chemotherapy drugs we use to kill cancer. Thymosin beta-4, this is the protein we are testing to see if it makes the treatment better. We used trypan blue for viability. This picture was taken in Qatar University lab. We were testing to see how many cells are alive and how many cells are dead after the treatment. And we used flow cytometry for the ROS analysis. This is important because if we identify molecules like TB4 that change how cells respond to stress, we can make chemotherapy more effective and less toxic. Chemotherapy results: the drugs cisplatin or vincristine work to kill cancer cells. Adding TB4, when we added thymosin beta-4, more cancer cells died. Stress levels: chemotherapy increased the amount of stress. Adding TB4 didn't change these stress levels. Gene changes: the study showed that these treatments change the genes that tell the cell to die or to handle stress. Our results are very promising for future cancer research. We want to use these findings to create a better and safer treatment that help more children survive medulloblastoma. Finally, we want to thank the High School Research Experience Program, QRDI, and the QU Health Research Office for their invaluable support and guidance. In conclusion, by using TB4, we aren't just making chemotherapy harsher, we are making the cancer cells weaker. Thank you for your time.